Inside every single human cell right now, billions of microscopic biochemical reactions generate metabolic energy, cellular building blocks, and inevitable cellular waste. Over time, damaged structural proteins, misfolded enzymes, oxidized cell membranes, and exhausted mitochondria accumulate inside your tissues, driving the fundamental biological process we recognize as aging. Fortunately, human cells possess an ancient, built-in self-cleaning system known as autophagy, a Greek term that literally translates to self-eating. First described by Nobel laureate Christian de Duva in 1963, autophagy was illuminated at the molecular level by Japanese biologist Yoshinori Ohsumi, who earned the 2016 Nobel Prize in Medicine for his groundbreaking discoveries. During autophagy, cells construct double-membrane vesicles called autophagosomes that engulf cellular debris, misfolded proteins, and damaged organelles throughout the cytoplasm. These autophagosomes then fuse with digestive lysosomes, utilizing powerful hydrolytic enzymes to break down toxic waste into raw amino acids, fatty acids, and nucleotides. The cell then recycles these newly liberated building blocks to repair cellular structures, synthesize fresh enzymes, and generate vital metabolic energy. When autophagic recycling functions at peak efficiency, your organs remain resilient, systemic inflammation stays controlled, and cellular energy production operates smoothly. However, as humans age, natural baseline rates of autophagy progressively decline, allowing senescent waste and oxidized cellular debris to build up in tissues over time. This age-related drop in autophagic capacity is directly implicated in neurodegenerative decline, cardiovascular stiffness, metabolic dysfunction, and loss of muscle quality. For decades, researchers believed that prolonged starvation, extreme caloric restriction, or intense endurance exercise were the only reliable triggers for systemic autophagy. While fasting is indeed a potent physiological trigger, modern longevity researchers have discovered a natural dietary polyamine compound capable of activating cellular cleanup directly: spermidine. Spermidine was originally discovered in 1678 by Dutch microscopy pioneer Antonie van Leeuwenhoek, who observed tiny crystalline structures forming in human semen samples under his early lenses. For over 3 centuries, spermidine remained an obscure biochemical footnote until modern molecular biologists uncovered its indispensable presence in every living plant and animal cell. Polyamines like spermidine, putrescine, and spermine are small, positively charged organic molecules that bind directly to DNA, RNA, and cell membranes to regulate growth and protein synthesis. In human tissue, intracellular spermidine concentrations peak during early childhood and steadily drop as we age, mirroring the gradual decline of natural autophagic clearance. To understand how spermidine triggers autophagic recycling without requiring multi-day starvation, we must examine its molecular interactions with cellular nutrient sensors. Primary control over cellular growth and recycling is governed by a master protein complex known as the mechanistic target of rapamycin, or mTOR. When amino acids and glucose are abundant, mTOR remains highly active, instructing the cell to build new proteins and grow while actively suppressing autophagic processes. Conversely, when nutrient availability drops or specific signaling molecules interfere, mTOR activity decreases, allowing the cell to switch into repair, maintenance, and autophagic cleanup. Intriguingly, spermidine acts as a natural caloric restriction mimetic, inducing autophagy through a unique epigenetic mechanism that bypasses direct mTOR inhibition. Specifically, spermidine inhibits an essential intracellular enzyme known as EP300, a histone acetyltransferase that normally keeps autophagic gene transcription suppressed. By inhibiting EP300, spermidine induces hypoacetylation of key autophagic proteins, signaling the cell to transcribe autophagy-related genes and assemble autophagosomes immediately. Because spermidine stimulates cleanup through EP300 inhibition rather than aggressive mTOR suppression, it avoids the muscle loss and metabolic stress, sometimes associated with severe calorie deprivation. One of the most vital targets of spermidine-driven cleanup is the selective clearance of damaged and dysfunctional mitochondria, a specialized process called mitophagy. Mitochondria serve as the cellular powerhouses, converting dietary nutrients and oxygen into chemical energy in the form of adenosine triphosphate, or ADP. However, as mitochondria age and suffer oxidative injury, they begin leaking harmful reactive oxygen species and pro-inflammatory signaling molecules into the cell cytoplasm. Mitophagy selectively isolates and digests these compromised mitochondrial units before they can trigger programmed cell death or tissue inflammation. By replacing old, leaking mitochondria with newly synthesized, efficient organelles, spermidine helps preserve cellular energy dynamics and reduces chronic oxidative stress. In brain tissue, autophagic dysfunction allows toxic protein aggregates like beta-amyloid, tau, and alpha-synuclein to accumulate in neurons, contributing to neurodegenerative disorders. Preclinical animal studies demonstrate that spermidine supplementation promotes the autophagic clearance of these misfolded brain proteins, supporting synaptic plasticity and memory retention. Neuroscientists observed that aging mice given spermidine exhibited superior spatial learning, faster maze navigation, and reduced markers of neuroinflammation compared to control groups. In cardiovascular science, spermidine has been shown to enhance nitric oxide bioavailability within endothelial cells, supporting blood vessel dilation and reducing arterial stiffness. Furthermore, spermidine protects heart muscle cells by maintaining healthy cardiomyocyte mitochondrial density and preventing pathological cardiac hypertrophy in response to high blood pressure. Cardiologists noted that spermidine-treated animal models maintained youthful left ventricular elasticity and experienced significantly lower rates of heart failure during aging trials. In 2009, a landmark study published in Nature Cell Biology by Professor Frank Medeo demonstrated that spermidine supplementation extended lifespan across yeast, fruit flies, nematodes, and human immune cells. Subsequent mammalian experiments confirmed that dietary spermidine administration reduced markers of arterial stiffness, preserved cardiac function, and enhanced memory performance in aging mice. Clinical interest in spermidine grew significantly following long-term human population studies examining dietary intake patterns over multi-decade periods. A major 20-year prospective study, led by researchers at the Medical University of Innsbruck, tracked over 800 human participants, evaluating their precise polyamine consumption. The findings revealed that individuals in the highest third of dietary spermidine intake experienced significantly lower overall mortality rates compared to those in the lowest third. Researchers calculated that high dietary spermidine consumption correlated with a survival advantage equivalent to being roughly 5 to 7 years younger biologically. While clinical trials continue to investigate optimal therapeutic dosages, nutritional scientists have identified the primary whole food sources highest in natural spermidine. Wheat germ is by far the most concentrated dietary source of spermidine, followed closely by aged cheeses such as Parmesan, cheddar, and Gouda. Fermented foods like Japanese natto, traditional soy sauce, and sauerkraut contain high polyamine levels produced through bacterial fermentation processes. Vegetables such as shiitake mushrooms, green peas, broccoli, cauliflower, spinach, and asparagus also deliver meaningful amounts of natural spermidine within a balanced diet. In addition to dietary intake, your gut microbiome synthesizes a portion of your systemic polyamines, provided your gut contains diverse strains of beneficial bacteria like lactobacillus. Probiotic formulations and fiber-rich diets nourish these polyamine-producing microbial strains, enhancing endogenously generated spermidine levels within the digestive tract. Cooking, boiling, and heavy industrial processing can diminish food polyamine levels, making fresh, minimally processed whole foods the most reliable nutritional source. Longevity scientists emphasize that while spermidine is a powerful molecule for cellular maintenance, it works best when integrated into a comprehensive healthy lifestyle. Consistent physical exercise, adequate slow-wave sleep, stress management, and nutrient-dense eating remain the indispensable foundation for metabolic health and natural autophagy. Combining nutrient-dense whole foods with regular physical movement creates a powerful synergistic effect that optimizes cellular cleanup and mitochondrial efficiency. As research continues to unravel the molecular intricacies of polyamines, spermidine represents an exciting bridge between daily nutrition and cellular longevity science. Supporting your body's built-in recycling systems through targeted nutrition may prove to be one of the most effective strategies for maintaining health span and biological youth. Remember that this video is for general informational purposes only and does not constitute medical advice. Always consult your physician or qualified healthcare provider regarding dietary changes or supplement protocols. Thank you for watching Calorierecipe. If you found this deep dive into cellular health valuable, please like this video, subscribe to the channel,